Hey guys, I am making this video to put on the announcements and hopefully it will help you be able to get through this week's lab materials. So hopefully this helps. Just a reminder, this is the last week for full labs, however you want to look at it. Next week, you'll be working completely on your final project. So those will be submitted next week. You'll need to submit your presentation and your paper. All right, so to get going, a couple of things. Let me just move some stuff over. It's a little slow, sorry. All right, so I'm going to actually put this into a PowerPoint that you can read from the beginning. So your lab this week, I'm going to do this lovely PowerPoint for you, and then we'll go over what you're supposed to be doing. So we're working on pulmonary ventilation. So a couple things about it is that we're talking about definitions. It's important to remember the term respiration, which refers to the liberation of energy from different pieces, from glucose, from fatty acids, from amino acids to form ATP at the cellular level. So then you have cellular respiration, which is something that we all need to do, otherwise we don't breathe. So what is commonly referred to as the function of respiratory system is external respiration, more appropriately called ventilation. So that's what we're working with, where it's the process of blood becomes oxygenated and released, and releases carbon dioxide through the lungs. So this tissue demand for oxygen depends upon what you're doing. So it can be different if you're sleeping, if you're running, if you're exercising, if you're sitting in a chair like we are right now watching this video. So those changes in respiration depends upon the outside stimulus. And so it reflects the tissues that demand oxygen. So another important function of ventilation is the release of carbon dioxide, which is the waste product of the cellular respiration, which is closely associated with the respiratory system in terms of pH homeostasis. So, oh, that's nice. Okay, so some of the things that we look at when we do this lab, generally when we're in the lab itself, is we're looking at volumes and capacities. So the spirometer is the study of the lung. Spirometry is the study of lung ventilation, sorry about that, which is a record of the air movement in and out of lungs. And we are using something called a spirogram to be able to visualize that movement. And we use a device called the spirometer. So when we're looking at something, or this particular thing right here, we're looking at the different points on the diagram, and we are boiling it down to volumes and capacities. So we're looking at things like tidal volume, which is what you normally breathe in and out as you're sitting here watching this video. You're breathing in and out, and that's your tidal volume. It's this line right here. And then depending on your activity either way will depend upon capacity of lungs or the volume of air that you're taking in. Okay, so if we're looking at volumes, the first thing we look at is the tidal volume, because that's the normal amount of air that you're taking in and out as you're breathing. So it's this one right here, this area right here. It'll come into play when we're trying to find out capacities later. We have the IRV, which is the volume of the air that can be inspired above the normal tidal volume. So it's this piece right here. And then you have the ERV, which is the volume that can be forced out after tidal volume, which is right below it. So you have your normal, and then above the tidal volume that can be inspired after, and then anything that can be forced out is below. The residue volume is the volume of air that the lungs remaining in the lungs after that forceful expiration. And so that helps to diffuse the gases as well between some of those pieces in the lungs. Okay, so if we're looking at volume, we're also looking at capacity, which is what this piece also shows, these pieces down here. So what we're doing is basically taking some of these pieces here and adding either those IRVs or the ERVs to get our how much air or how much lung capacity we actually have. So IC, in order to create that amount or to figure out what our IC is, or the inspiratory capacity, you're taking your tidal volume, which is right here, and then you're adding that IRV, which is this part here. So if I were measuring that, I would take the bottom here of the tidal volume all the way up to the top of the IRV, and then that would give us the IC capacity. Functional residue, it consists of the expiratory reserves, which is in the bottom part here, and this residual volume. So that piece from this mark here where my piece, or where my, oops, sorry, where this is down to the bottom of that residual volume piece would give us our capacity there. Vital capacity is the volume of air expired from the maximal inspiration to the maximal expiration. So basically this calculation, so VC equals the IRV plus the tidal volume plus the expiratory. So this whole section right here is what we would use to do vital capacity. And if you look over here, it actually has that line to show you what you're adding up. So you would add up every single one of those sections to get that vital capacity. And then total lung capacity, obviously we're adding in the entire piece or lung volume measurement all the way from the top of the inspiratory to the bottom of the visual or the residual, which is this entire section here. Okay, so a couple other things. Remember that a spirometer is a device that we use to measure those lung volumes. That's what we're using, or we normally would use in the lab. Many breathing disorders can be detected by doing this. So if we find pneumonia or pulmonary restrictions or bronchiogenic carcinoma, pulmonary congestion, possibly even things like coronavirus that we're seeing right now. And then of course, if we're looking at something, an example of that would be emphysema. So emphysema will cause the tidal volume to increase and the IRV to decrease. So you're going to have that increase. If you go back to this, emphysema, you see this increase and this area up here decrease. So that gives an indication that emphysema is there. All right, a couple other pieces. Just respiration in general. It basically gives us everything to get rid of oxygen, or to utilize oxygen and get rid of carbon dioxide into those 5 stages. So pulmonary ventilation, the movement of gas between lungs and the environment, gas exchange, which is the movement of gases between the lungs and the bloodstream, and then gas transport, which is the movement of gases within the bloodstream, tissue blood gas exchange. It's basically the movement between the bloodstream and the tissues that you have. So cellular respiration obviously is the consumption of oxygen to produce carbon dioxide by cells and organelles, which we hopefully remember from our last biology piece. All right, a couple other things. That human respiratory system consists of tubes within the lungs, and during pulmonary gas exchange, oxygen and carbon dioxide cross between those two things and diffuse through respiratory membranes, and then those have a 2-cell layer thick, those epithelial, I can never say this word, the E cells of the capillary. So hypercapnia, just so that you know, and hypoxia are two conditions that we look at. Hypercapnia is the condition of too much carbon dioxide in the blood, and then hypoxia is the condition where you have not enough oxygen in the blood. So those are just a few other odds and ends that we look at during this lab for pulmonary. All right, so one more thing that I would like to do. Let's see. All right, just a reminder that you still have your post-lab, pre-lab, all that good stuff. In the announcements this week, I have included some important information. So you'll still have your post-lab, you're still going to be doing pre-readings, all of that good stuff. Those things are all due by Friday, by Saturday the 18th. And then your quiz is available from Thursday to Saturday. So make sure that you have taken that quiz before that time. As always, if you have questions, if you have questions or you need help, you can email me at any time. And I hope you guys are doing well and staying safe. Have a good week, and we'll see you next week. good luck on your presentations. I look forward to seeing all the cool things you guys have done.